Hello everyone, in today's class we'll try to learn about body fluids. The objectives of this class would be to give normal values of total body water as a percentage of body weight in different age group in male and females, give the percentage of distribution of body water in different body fluid compartments, understand the concept of water gain and water loss and list the factors that influence water balance, explain the effective circulating volume and its application, and also name the different body fluid compartments, total fluid present in the compartment, and composition of ions in each compartment. Give the characteristic of ideal indicator used to measuring the body fluid volumes. Name the units of measurement. Name the indicator used. Give the basic principle and method of measurement of body fluid volume of different compartments. Explain why dehydration is common and often severe in children. Stability in the volume and composition of body fluid in the body fluid compartments is essential for homeostasis. Functions of cells depend on fluid both outside inside the cells. Therefore, maintenance of fluid volume of the body is important aspect of patient management in clinical medicine. Appropriate fluid replacement in dehydration and removal of fluid in overhydration, hypervolemia, and edema are mainstay treatment of the fluid volume disorders. Coming to body fluid content, the body composition of normal adult male is on average consists of 50% of fat, 18% of proteins, 7% of minerals, and 60% of water. The total body water as a percentage of body weight on an average is about 60% in adult males, which is about 10% more than adult females. In infants and children, though the total body water as a percentage of body weight is more than adults, their absolute water content is significantly less. In elderly people with both sexes, total body water is about 10% less than the adult counterparts. In females, total body water is less than male due to relatively greater amount of adipose tissue, that is subcutaneous fat in them. Of total body water, about two-thirds is present in the intracellular compartment and one-third is in extracellular compartment. So this gives the total body water as a percentage of body weight in males and females at different ages. Okay, so it is going to range from 65% to 42%. Okay, so in infants, children, in adults, it is high compared to the elderly age group. So this is the distribution of total body water into three compartment, that is total body water, uh, extracellular volume, and uh, intracellular volume. So total body water can be divided into two parts, that is extracellular fluid volume and intracellular fluid volume. So out of 60% of the total body weight, 40% is ICF and 20% is ECF. So it can be given as 20, 40, and 20 rule. Normally, volume of the different body compartments can be calculated by following formula. So total body water is equal to zero point 0.8 into body weight intracellular fluid, zero point 0.4 into body weight extracellular fluid, zero point 0.2 into body weight interstitial fluid volume, 0.75 into ECF, and plasma volume is equal to 0.25 into extracellular fluid volume. The body fluid volume depends on water balance. The control of volume of body fluids directly linked with the water balance of the body. Therefore, before discussing the regulation of the body fluid volumes, first let us understand the mechanism of water balance. Normally, water balance is achieved by matching the water gain with water loss. The body gains water from two sources, exogenous and endogenous. Exogenous water gain occurs mainly by two processes, drinking of the water and water ingested with the food. The exogenous water intake normally ranges from 2 to 10 liters per day. Drinking of water is a major form of water gain. It ranges from zero point 0.5 to zero point 0.8 liters per day, depending mainly upon the environmental conditions. Water intake is mainly controlled by the thirst center in the brain. Water is present in all the food preparation and fruits. On an average, water ingested in food, that is in breakfast, lunch, snacks, and dinner, varies from 1 to 5 liters per day. Water is one of the product of metabolism. The other products are ATP, carbon dioxide, and increase in the temperature. During process of tissue oxidation, water is continuously being produced in the body. Endogenous production of water accounts for about 300 to 500 ml of water uh, added per day to the body fluids. Water loss from the body occurs mainly in four forms: urine, feces, sweat, and insensible loss. Urinary loss of water is a major form of water output from the body. About 1 liter of water is excreted from the body in urine per day. Kidney plays important role in water balance by altering water excretion in urine, which primarily depends on the concentration of ADH in the plasma. Normally, about 200 ml of water per day is excreted in the feces. Sweating is greatly influenced by environmental temperature and level of physical activity. The rate of sweat production per day is about zero point 0.1 liter at ambient temperature of 23 degrees Celsius, 1.5 liter at 38 degrees Celsius, and 5 liter at 45 degrees Celsius. Water is continuously evaporated from the skin surfaces, which is called as insensible water loss. Water is also continuously evaporated from the respiratory passage in breathing. What is positive and negative water balance? Positive water balance occurs when water gain is more than its loss. This causes hemodilution, therefore decreases the osmolality of the plasma, decreases the red cell count, and produces hyponatremia. Negative water balance occurs when water loss is more than its gain. This leads to dehydration and causes hemoconcentration. It increases plasma osmolality. Due to hemoconcentration, there is a relative increase in RBC count. Many factors which influences the body water, that is plasma osmolality, is one. The plasma osmolality is an important factor for water balance. Alteration in the osmolality is detected by osmoreceptors in the hypothalamus that stimulates the thirst. Increased plasma osmolality is also primary stimulus for ADH secretion, and the factors that affect the plasma osmolality are it mainly depends on the osmotically active molecules in the plasma, especially sodium chloride, proteins, and glucose. Alteration in blood volume is detected by volume receptors in atria and send impulses to hypothalamus and to pituitary to secrete ADH. However, increase in osmolality is a better stimulus than hypovolemia for stimulation of thirst and ADH secretion. Factors that affect blood volume are the plasma volume and the hematocrit. Volume sensors. Volume sensors or the extracellular volume sensors are the volume receptor located in the vascular tree that responds to stretch of the vessel wall. Generally classified into two categories: low pressure baroreceptors and high pressure baroreceptors. Low pressure volume receptors are located in low pressure side of circulation, especially atria and in pulmonary vasculature. High pressure volume receptors are present in high pressure compartment of circulation. So these receptors include baroreceptors present in the carotid sinus and arch and left ventricle. Coming to body fluid compartments, so there is about 42 liters of body fluid in adult male, which is about 60% of the body weight. The body fluid distributed mainly between extracellular compartment and intracellular compartment. The extracellular compartment is the fluid in the spaces outside the cell constitutes extracellular fluid. This includes the plasma and the interstitial fluid. The ECF constitute about 20% of the body weight, which is about 14 liters. So this is a diagram showing the distribution of the body fluids. Um, so this is the total body water. In that, this is the intracellular uh, almost occupies two-third, and extracellular one-third. The intercellular, I mean, if the total body water is 42 liters, intercellular fluid is 28 liters, extracellular fluid is 14 liters, plasma is 3 liters, and interstitial uh, fluid is around 11 liters. The exchange of the fluid from the environment, it mainly occurs through the plasma, and then plasma fluid is exchanged with the interstitial fluid, and then interstitial fluid is exchanged with intercellular fluid. So same thing, the the distribution of ECF, I mean, if you see, the 25% is the plasma and 75% is the interstitial fluid, so which is around 3.5 liters and 10.5 liters respectively. Intracellular fluid compartment is the fluid contained within the cells of the body. It constitutes about two-thirds of the total uh, fluid of the body. It cannot be measured directly. So it is measured by subtracting ECF volume from the total body fluid volume. Transcellular fluid is present in this fluid is present in another compartment of the body called as transcellular fluid. I mean, apart from the extracellular fluid volume, it is a, and also it is a part of extracellular fluid volume. So, which represents the body fluid present in the lumen of structures lined by the epithelium. It includes fluid in the secretion of exocrine digestive glands, cerebrospinal fluid, pleural, peritoneal, synovial, and pericardial fluids, and intraocular bile, luminal fluids of the gut, and fluid in the cochlea and kidney tubules. The total transcellular fluid volume is around 1.5% of the total body weight, which is about 1 to 2 liters. How to measure the body fluid volumes? So, general principle utilized here, the volume of the fluid compartment usually measured by the indicator dilution principle, so which is based on the following relationship. The concentration of any substance can be given by C is equal to A divided by V. So where A is the amount of dye which has been present or injected intravenously, uh, and the volume is the amount of the volume in which the dye has been distributed. Okay, so where A is amount of substance injected intravenously, and V is the volume in which the substance is distributed, and C is the final concentration. Okay, so if we can modify this equation, so the volume becomes the average concentration of dye divided by concentration of that substance uh, uh, in that compartment. So this formula can be used for measurement of the body fluids. So some of the indicators which are used for the measurement of body fluid compartments, they should have a certain characters, should be relatively easy to measure, should remain in the compartment which is being measured, uh, should not change the fluid distribution in the compartment being measured, should be non-toxic, must mix evenly throughout the compartment being measured, should remain unchanged by the body during mixing period, or the amount changed must be known. The size of the body fluid compartment is measured by injecting substance and is then calculating the volume of fluid in which the test substance is distributed. So this is called as volume distribution of the injected substance, which is equal to the amount of uh, dye injected divided by the concentration of that substance in the sample. Okay, so this is the formula is used. If the dye leaves the compartment by excretion or metabolism during the time allowing mixing, then the calculation is done as follows: volume distribution is equal to amount of dye injected and amount of dye or substance removed divided by the final concentration of that particular substance in that body fluid compartment. So, methods of measurement. The ECF volume is measured by volume distribution principle using the inulin, a polysaccharide having a molecular weight of around 5200. So even radioactive inulin is prepared by substituting 14C for one of the carbon atoms of the molecule. Radioactive inulin levels are easily determined by the counting the samples with the suitable radiation detectors. And also chloride 3638, bromide 82, mannitol, sucrose are also used for measurement of ECF volume, because these substances they get distributed only in the extracellular fluid volume. The chloride is largely extracellular, therefore radioactive isotopes chloride are used for the purpose. However, the ECF volume determined by using chloride is greater than actual volume, as some chloride is also present in the intracellular fluid. 82 bromide, sulfate, thiosulfate, thiocyanate, ferrocyanide are also used for measuring ECF volume, as these ions interchange with the chloride in the body and they determine the greater values for ECF. Mannitol and sucrose have also been used to measure the ECF volume. Measurement of plasma volume. There are two dilution methods. The first one, the substance used uh, neither leaves the vascular system nor penetrates red cells. For example, Evans blue dye and radioiodinated human serum albumin, that is RISA, and radioiodinated gamma globulin and fibrinogen. Second method, the radioisotopes of phosphorus, iron, and chromium are used that penetrate and bind to red cells. Commonly used tag is the chromium 51, which is attached to the red cell by incubating in a suitable chromium solution. Then plasma volume is calculated by using formula. The plasma volume is equal to 100 minus packed cell volume divided by 100 into blood volume. Measurement of the interstitial fluid volume. The interstitial fluid volume is estimated as the difference between ECF volume and plasma volume. The intracellular fluid volume cannot be measured directly by dilution principles, as no substance remains confined to only to this compartment. It is, it is determined indirectly by subtracting ECF volume from total body water. So thus first total body water is measured. The measurement of total body water is performed by indicator dilution principles. Deuterium oxide, or it is also called as heavy water, is usually used as uh, it has a properties that is slightly different from water. Tritium oxide, aminopyrin can also be used for measuring total blood volume. Since these substance they are going to be distributed in the all the body fluids. So this is in brief about the body fluids, how they are differentiated and how to measure the different body fluid volumes. Thank you.